In this tutorial, we are going to learn about genetic transfer and recombination in bacteria, also known as plasmid. Genetic recombination. It means transfer of DNA from one organism to another recipient. The transferred donor DNA may then be integrated into the recipient's nucleide by various mechanisms, either homologous or non-homologous. A. Transformation, B. Transduction, and C. Conjugation. Transformation. Transformation means the transformation refers to the transfer of relatively small segment of naked DNA from a donor cell competent recipient cell, that is, male to female. Bacterial transformation was first discovered by Griffith in 1982 between the two strains of Streptococcus pneumonia, which causes pneumonia in humans and mice. Transformation involves 4 steps. Genetic recombination, in which a DNA fragment from a dead, degraded bacterium enters a competent recipient bacterium and it is exchanged for a piece of the recipient's DNA. The 4 steps in transformation. Step 1. A donor bacterium dies and is degraded. The dead bacterium, which has a dots body, it is a dead bacterium. And the fragments present are the DNA fragments of a dead bacterium. Step 2. A fragment of a DNA from the dead donor bacterium binds to DNA binding proteins on the cell wall of competent living recipient bacterium. Just look onto the screen. The dead donor bacterium binds to DNA binding proteins of the female bacterium. Point 3. The Rec-A protein promotes genetic exchange between a fragment of donor's DNA and the recipient's DNA. Step 4. Exchange is completed. B. Transduction. Genetic recombination, in which a DNA fragment is transformed from one bacterium to another by a bacteriophage, is known as transduction. What is bacteriophage? Bacteriophage means the virus carrying the bacteria. On the screen, you can see the structure of T4 bacteriophage. On the anterior side of bacteriophage, head or capsid is present, and in the middle, the tail is present, and at the base plate and tail fibers are present. History of transduction. The phenomenon of transduction was first discovered by Zinder and Lidenberg in 1952. They discovered this while searching for sexual conjugation in Salmonella species. What are bacteriophages and how they survive? Bacteriophage or a phage are obligate intracellular parasites that multiply inside bacteria by making use of small or all the host biosynthetic machinery, that is, viruses that infect bacteria simply. And what are obligate parasites? Obligate parasites need a host to complete its life cycle. There are 2 types of transductions: generalized transduction and specialized transduction. In generalized transduction, a DNA fragment is transferred from one bacterium to another by a lytic bacteriophage that is now carrying a donor bacterial DNA due to an error in maturation during the lytic life cycle. And second is specialized transduction. A DNA fragment is transformed from one bacterium to another by a temperate bacteriophage that is now carrying a donor bacterial DNA due to an error in spontaneous induction during the lysogenic life cycle. 7 steps is in generalized transduction. Step 1. A lytic bacteriophage adsorbs to a susceptible bacterium. Point 2. The bacteriophage genome enters the bacterium. The genome directs the bacterium metabolic machinery to manufacture bacteriophage components and enzymes. And in point 3, occasionally bacteriophage head or capsid assembles around a fragment of donor bacterium's nucleoid or around a plasmid instead of phage genome by mistake. Point 4. The bacteriophages are released. Point 5. The bacteriophage carrying the donor bacterium's DNA adsorbs to a recipient bacterium. Point 6. The bacteriophage inserts the donor bacterium's DNA it is carrying into the recipient bacterium. Point 7. The donor bacterium's DNA is exchanged for some of the recipient's DNA. Bacterial conjugation. Bacterial conjugation is genetic recombination in which there is a transfer of DNA from a living donor bacterium to a recipient bacterium, and often it involves a 6 pilus. For the first time, Lederberg and Tatum in 1946 presented the evidence for bacterial conjugation, that is, a process of transfer of genetic material by cell-to-cell contact. The three conjugative processes are F plus conjugation, HFR conjugation, high-frequency conjugation, and R plasmid conjugation, that is resistant plasmid conjugation. F plus conjugation, the genetic recombination in which there is a transfer of an F plus plasmid but not chromosomal DNA for a male donor bacterium to female recipient's bacterium. It involves a 6 pilus other plasmid present in the cytoplasm of the bacterium, such as the coding for antibiotic resistance may also be transferred during this process. F plus conjugations involve 4 steps. The 4-stepped F plus conjugation. Step 1. The F plus male has an F plus plasmid coding for a 6 pilus and can serve as genetic donor. Step 2. The 6 pilus attaches to an F minus female recipient. One strand of the F plus plasmid breaks. Step 3. The 6 pilus retracts and a bridge is created between the two bacteria. One strand of the F plus plasmid enters the recipient bacterium. Step 4. Both bacteria make a complementary strand of the F plus plasmid and both are now F plus males capable of producing a 6 pilus. There was no transfer of donor chromosomal DNA, although other plasmids the donor bacterium carries may also be transferred during F plus conjugation. HFR conjugation or high frequency of recombination. Genetic recombination in which fragments of chromosomal DNA from a male donor bacterium are transferred to a female recipient bacterium following insertion of an F plus plasmid into the nucleoid of the donor bacterium involves a 6 pilus. And the HFR recombination involves 5 steps. 5-stepped HFR conjugation. Step 1. An F plus plasmid inserts into the donor bacterium's nucleoids to form an HFR male. Step 2. The 6 pilus adheres to an F minus female recipient. One donor DNA strand breaks in the middle of the inserted F plus plasmid. Step 3. The 6 pilus retracts and a bridge forms between the two bacteria. One donor DNA strand begins to enter the recipient bacterium. The two cells break apart easily so that only a portion of the donor's DNA strand is usually transferred to the recipient bacterium. Step 4. The donor bacterium makes a complementary copy of the remaining DNA strand and remains an HFR male. The recipient bacterium makes a complement strand of the transferred donor DNA. Step 5. The donor DNA fragment undergoes genetic exchange with the recipient bacterium's DNA. Since there was a transfer of some donor chromosomal DNA, but usually not a complete F plus plasmid, The recipient bacterium usually remains F-. Resistant plasmid conjugation, genetic recombination in which there is a transfer of an R plasmid from a male donor bacterium to a female recipient bacterium, involves a sex pilus. Resistant plasmid conjugation involves 4 steps. Step 1. The bacterium with an R plasmid is multiple antibiotic resistant and can produce a sex pilus. Step 2. The sex pilus adheres to an F- female recipient. One strand of the R plasmid breaks. Step 3. The sex pilus retracts and a bridge is created between the two bacteria. One strand of the R plasmid enters the recipient bacterium. Step 4. Both bacteria make a complementary strand of the R plasmid and both are now multiple antibiotic resistant and capable of producing a sex pilus.